Hi, it's Tom with Biology Magnets. This video is over Module 10B, MAPK signaling pathways. And I've got it set up here. We start with these epidermal growth factor molecules. These are the ligands that are going to bind to receptors that are on a cell membrane. And what this whole thing is going to do is going to cause the cell to multiply eventually. That's why it's called MAPK. MAPK stands for mitogen activated phosphate kinase, which a mitogen is a molecule that causes mitosis, that causes cells to multiply. So these are considered to be mitogens. Epidermal growth factor is the name of these proteins. And they're received by proteins in the membrane. This is a membrane I've drawn with markers. The epidermal growth factor is received, the signal is received by these epidermal growth factor receptor molecules that are in the membrane. Now they're usually separate, but once they receive the signal, they'll actually move together and form what's called a dimer. Dimerization is what they call that. And this causes changes internally. What's going to happen is an ATP molecule, that's what this is, ATP, is going to phosphorylate the bottom part of these receptor proteins, which have these tyrosine residues that will actually pick up a phosphate from ATP, and they'll actually phosphorylate the tyrosine residue on the opposite side. And that happens on this side too. And what can end up happening is some or all of these tyrosine residues can be phosphorylated. And so that happens once these epidermal growth factors are received. So these come together, you have the phosphorylation, and then you'll have a protein called GRB2 that can connect to one of these phosphates and the growth factor receptor and bind to it. Another protein comes up called SOS, son of sevenless is the name of it. It's an interesting name, but it comes from proteins in Drosophila, and the name comes from them. And son of sevenless will bind to GRB2. And then that complex can associate with this RAS protein. Now the RAS protein is a type of G protein. The reason we call it a G protein is it's associated with a GDP molecule, guanosine diphosphate. And the GDP molecule is attached to this RAS protein, and it's in its off position. But this protein will come in here, and I want you to notice it has a lipid tail. The lipid tail helps keep it in the membrane. So this RAS protein is associated with the membrane. But the RAS protein will slide over here and will bind to the son of sevenless. And that changes the configuration of the RAS protein so that the GDP molecule falls out and is replaced by a GTP molecule, guanosine triphosphate. And that changes the formation of the RAS protein where it can become active. So we've got the reception of the signal and then a bunch of things going on inside the membrane. Now, the RAS protein, after it loses the GDP and is replaced with the GTP, will come loose and will move along the membrane and will associate with another molecule in the cell called BRAF. Now this is a protein here that is a kinase that can phosphorylate other molecules. So the BRAF becomes phosphorylated by ATP, will drop a phosphate off and phosphorylate the BRAF. And then the BRAF will in turn phosphorylate other molecules. We have what's called here a phosphorylation cascade, where BRAF is phosphorylated after binding with the RAS protein. And then the proteins called MEK, there's actually two of them, MEK1 and MEK2. But I've got them together as one magnet here. But anyway, there is a spot on the BRAF where an ATP can come in and attach to the BRAF molecule, and that can cause phosphorylation of the MEK1 and 2. And then the MEK1 and 2, another ATP can bind with that. There's a spot for the ATPs to bind right here. This edge of the molecule here. And the MEK1 and 2 can phosphorylate this ERK1 and 2 molecule. So we have BRAF phosphorylating MEK1 and 2, MEK1 and 2 phosphorylating ERK1 and 2, and that can just continue to go on. The ERK1 and 2 can phosphorylate. Here's another ATP coming in. It can attach to the ERK1, 2 molecule. There we go. And this ERK1 and 2 can phosphorylate this transcription factor. It's called Phos-Jun. And once the Phos-Jun transcription factor is phosphorylated, it can enter the nucleus and bind to the DNA in the nucleus. And we can see it bind there. It fits nicely. And this DNA, this gene is called AP1. And that gene, the binding of this transcription factor, this Phos-Jun transcription factor, causes the AP1 gene to be transcribed and translated, and that will make proteins that cause the cell to divide. So when we talk about cell signaling, we talk about three parts. The reception of the signal, the transduction is the moving of the signal to the nucleus, and then the response. Reception, transduction, and then the response in the nucleus making proteins for cell division. So this can be regulated. There is a protein that can regulate this to some extent. It's called this GAP protein. And the GAP protein can stick to this RAS protein. There's a place for it to stick here. And when it does, it causes this last phosphate on the GTP to come off. And when this phosphate comes off, it changes the configuration of the RAS protein, and the BRAF will no longer be able to stick to it. And that will stop the whole phosphorylation cascade. So this GAP is a protein that can regulate how much this occurs and how often we're telling our cells to reproduce. As a matter of fact, a mutation in the RAS protein that does not allow the GAP protein to bind is one source of cancer, where if this can't bind, we can't shut this down, and that can lead to cancer. So that's something that a lot of the biology texts discuss, this MAPK signaling pathway. There's one other thing that's included in this kit is simply an estrogen receptor and an estrogen molecule. If you want to talk about steroids, the way steroids work, the steroid molecule, this is estrogen, can come right through the membrane and attach to this estrogen receptor. And when it does, let me move this out of the way. When it does, that can enter the nucleus and bind to the DNA. It doesn't bind to the AP1 protein, but it can bind to DNA in various locations and cause responses. So that's how steroids work. Steroids come through the membrane because they're fat lipid based, and can go all the way, either be received in the cytoplasm or received in the nucleus. They can go right through the nuclear membrane too. And that can cause a transcription factor to be activated, to bind to the DNA, and cause some sort of response. So you can also talk about steroids with this lesson 10B. If you want more information or more detailed instructions, go to biologymagnets.com.